today for Olaris's inaugural seminar series. Truly super excited to see so many people and just have the opportunity to do this. I thought before we get started and before I introduce our panel, first give a little bit of context of why we're here. So Olaris is 8 years old, and we've had the pleasure of being located in many different spots throughout the state during our sort of career and trajectory. And each space that we were at was special and had its own sort of atmosphere and ecosystem. And I think I speak for myself and Chad and some of the team members that, you know, while we were there, we learned a lot of stuff. And I certainly started dreaming about what I would like the ecosystem to be at a place where Olaris could have its own spot and where we could call home. And so it's with that spirit in mind that we're trying to put together a series of events that will bring people together for collaboration, heated discussion, I hope, and also some good laughs. Because I think that like in science or in business, those like aha moments come from people being in the same room and sharing ideas, not necessarily a Zoom room either, and just sort of really getting into things about stuff that we're all passionate about. So with that, it is my absolute honor to welcome our panel to the stage up here. So come on in, guys, and we can all grab a seat. So we have assembled an expert panel of folks that are working in different omics. And how we thought we would structure the panel is first just ask a series of discussions and topics that we all sort of care about, and then invite the audience to participate. So to get us started, maybe Paola, I'll start with you. Could you give a quick introduction? Can you hear me okay?
Sure.
Okay. So I'm Paola Castaldi. I'm currently the Vice President of Clinical Biology and Proteomics at LifeMine Therapeutics. Within the company, we utilize several omics technologies, starting from genomics to transcriptomics, proteomics, metabolomics, to really predict which fungal strains will produce a new genetically encoded small molecules that will target therapeutic targets. And over the, before LifeMine, I spent more than 10 years in between large pharmas, AstraZeneca and Sanofi, where I mainly been focused on proteomics to understand the mechanism of action of our molecules, the target engagement, selectivity profiling, and really understand the diabetes of the molecules before they get into the patient.
I'm Eleanor Howe. I'm the founder and CEO of Biomedicine Data Science, and we're a consulting firm.
Hey Liz, do you have a microphone?
Yeah.
I'm an old guy. I can't hear you very well.
So I'm the founder.
Is it on? Is it on?
It looks on.
Okay.
Sounds awesome.
We're working on our AV here. This is the first one.
So I'm the founder and CEO of Diamond Age Data Science, and Diamond Age is a consulting firm that does computational biology, and we do data analysis for all of these omics that Paola was talking about. So genomics, transcriptomics, proteomics, a little metabolomics even, and then the data science and the statistics and engineering and infrastructure support that you do all this as well. So I'm a computational biologist by training. I started a company, and people depending on how you count them. It's different from day to day because we have contracts and we start up. We understand. And we help small companies do all of this data analysis.
Awesome. Thank you very much. Shourjo?
Can you guys hear me?
No, I can't hear you.
Why am I yelling?
I don't think the mic works.
Funny.
Yep.
It's
Good.
all right. Better?
Yeah.
Okay.
Sorry, guys.
No worries. Hello everyone, my name is Shourjo Ghose. I represent Bruker and Photonics. It's a part of the Bruker family where we specialize in, as a tools company, developing mass spectrometry techniques for, among other things, omics applications. My background is small molecules and proteomics, and I manage Bruker's proteomics business development team in North America. It's a pleasure to be here.
Are you in Villarica?
We are in Billerica.
Thanks.
I'm Andrew Geller. I work for Eurofins enterprise right across the street. We share space with Olaris and Liz. Eurofins Clinical Enterprise is a high throughput molecular testing laboratory. We've developed scalable high throughput sequencing, microarray, qPCR assays. So my job is to help look at the strategy the company developed, the technologies, and hand them off to a production environment where we can run testing for various customers from different institutes.
Well, thank you all again for joining. As you guys can see, we have very diverse sort of backgrounds here from, we'll say, genomics, proteomics. I didn't give my background, but I'm Liz O'Dan, CEO and founder of Olaris. We're a metabolomics diagnostic company. We have Eleanor taking in all of the data and trying to do analysis and statistics, and then Paola, who's using it for drug development. So omics can be a lot of things and can touch a lot of different areas. But I wanted to sort of start out by doing some level setting to make sure that we all sort of understand what we say or what we mean when we say the word omics and what each omics can tell us. So, Andrew, I'm going to put you on the spot first. When you and I were speaking about all the omics technologies, you had this great sort of comparison to a map and a topological landscape. Maybe you can help us get oriented through that.
Yeah. So I kind of thought of it, I mean, and this is just from the high level as a layered approach. I mean, you have foundationally genomics, which touches the foundation closest, and sitting on top of it, you have these various other areas, transcriptomics and expression, and then you move up into proteomics, metabolomics, and the way that I try and think about it is, how closely related is genomics to metabolomics. It's further in terms of its geographical relationship compared to, say, proteomics and metabolomics are. And you also bring in methylation as well, but instead of just sort of a spatial two-dimensional map, I kind of see it as a layered approach, and their relative relationship with one another is different depending on which omics space you're in. So I think that goes into pairing different types of omics for a specific purpose, right? And selecting what types of tests you're going to run for research or clinical trials. So that was, this was sort of just naturally us discussing it, and because I sometimes have to reorient myself. So.
Oh, I think it's good, and maybe sure I'll ask you to maybe expand. I think you had an additional way of viewing the omics as sort of filling gaps between one area.
Right, correct. So, I mean, all of us in this audience are aware of something wrong, right? If you think about genomics, I consider that as a hieroglyphic, something that is written in stone, which is very, very, it's like the blueprint of life, as some folks like to call it. Proteomics, on the other side, is fundamentally the study of the proteome, the proteins that we cell. That's typically a main target of today's drugs. Proteomics, if you consider that as an analogy, is the print media. The print media is an iteration of what is written in stone. It's got its own iterations, its own modifications based on the seed class of the environment, and it just gives you a snapshot of what the climate of that age is. With the lack of a better analogy, metabolomics and lipidomics is more like TikTok but Twitter. It provides you with a snapshot of what's happening at that moment. For example, if you take your lipid content profile in the morning when you get up, and then after having a cheeseburger, it is widely different. So in that way, it's an interplay between metabolomics, lipidomics, proteomics, genomics that helps us in cancer research, in developing drugs, developing therapeutic biomarkers that's going to be used for the research and eventually better patient outcomes that we're all viewed to provide.
Yeah, I think that's great. I mean, I don't know if I can take offense to the correlation between metabolites and TikTok, but I get the analogy that you're making, and I know we have some lipid people as well in the audience, but I think it is interesting how, and when I think of the different omics, I also think it's like number of touch points as well, right? So genomics is like a one snapshot in time, where as you maybe build up that foundation you were talking about, Andrew, like you get more and more touch points with the environment.
I mean, and also too, because it's so foundational, it can many times and in diagnosis, disease diagnosis, have the biggest impact. Biggest direct impact when you're talking about rare diseases and a million germline diseases, but at the same time, the majority of the genome is small variants with very minimal or no impact or unknown. So it can have a huge impact, a direct impact, but for the most part, it's kind of just the building blocks and your environment, you got all these other things. The other omics are impacting you directly on a day-to-day basis. And, overall health.
I think that's really important. And so sometimes, sometimes people think, like, they always ask me, how is metabolomics better than genomics? And you might get the same thing for proteomics. And I say it's not, but they're telling sort of different things, and genomics to me is like the first starting place of where you should kind of get set and then fill in the gaps.
Yeah, you can't build a building without a blueprint,
Yeah.
right? I mean, so you need to understand the genome, you understand the modifications, and then sometimes you pointed out there's not a direct correlation with phenotype, and that's where proteomics, metabolomics, and lipidomics come in. But to understand that, you have to backcalculate the genome and understand that these phenotypes, these phosphorylations, glycosylations in a particular peptide or a protein, does it have a correlation on the genome, and if so, how can we predict that? Fundamentally, it's one thing to cure diseases, but it's another thing to actually prevent that. And finding biomarkers that lead to a disease state early on is critical to therapy.
So I'm going to use that to parlay and ask a question for you, Eleanor. So we just talked about we can measure all these different omics, all these different data points. They can or cannot be correlated to different sort of disease states. So I'm sure as sort of our biostatistician on the stage here, we're going to turn to you for, I'd say, advice and maybe observations of what you're seeing as more and more people are now able to do omics studies. What are you seeing for good and bad trends, and like what should we be thinking about?
I think the thing that really distinguishes all these genomics and other technologies from each other is not so much which of them is more useful, but rather which of them is cheapest. Genomics it's pretty cheap. It's pretty cheap. It's not cheap, but it's way cheaper than proteomics. Let's face it, and it's also another technology that's established. It's always your first stop when you're investigating something because you can take a sample and send it to somebody and they'll do a easy job getting you a whole bunch of information back. A very straightforward way because you know how. Yeah. Proteomics and metabolomics really are the Wild West. It's totally different ballgame. It's more expensive. It's harder. The data is rarer. It's harder to get. And so there's a lot more new things we can learn out there. Right, so that's to me the thing that I'm seeing changing is a couple of things. You almost take genomics for granted. You have to do a good job with it, but it's like table stakes for good drug discovery and good diagnostics, of course. Transcriptomics, increasingly the same deal. The methods are established. It's not that expensive a technology. It's single cell. Proteomics, nobody knows what to do with. Mass specs are incredibly expensive, and the skill sets for analyzing the data is not very common, and the statistics are not as well established. All of that stuff. But it's really essential to figure out if that transcriptomic profile that you just got actually has a real effect on the cells. You need to check it at the protein level and see if there's something there. So that's a lot of the multi-omics work that we end up doing is that validation stuff. We don't do as much multi-omics with metabolomics, but it's the same deal. Is this change in the genome, is this change in the transcriptome, you're actually having a real impact on your drug or your patient. And that's where we end up combining the data.
Yeah, I think that's powerful. I think you bring up a good point that like genomics and transcriptomics are established and know how to handle the data sets, where in proteomics, I'll let you jump in, but I'll say for metabolomics, like we're still learning, and Amy's an expert in metabolomics as well, but how do you normalize your data properly, right? How do you make the most reliable cutoffs as real life examples. So when you and I were talking, you gave advice that I think is good about when you're starting a proteomics study, people that you might want to have around you.
Oh, I don't know if I'm going to remember exactly what I said before.
That's okay, I can help you out if not, because I could take it to heart. But.
Well, I probably said something about wanting, someone who really knew the technology and chemistry really well, so you did.
Yeah, so you said two people, that you should have a statistician, but not just anybody that knows stats, but someone that actually
smart. This is a great idea.
This is yours. But I think that's really important because, like, sometimes just applying out-of-the-box statistics to these technologies doesn't make sense because you don't know how maybe the signal amplifies over time and stuff like that.
Yeah, I have a great example of that. So one of the things, RNA splicing is one of the things that are great. You know, it's just different. You cannot assume anything about the distribution of utilization of splice sites in a given RNA. It's not a normal distribution. You cannot necessarily use a t-test to compare it to other things until the changes in splice site utilization. Big control if you're looking for a change in splicing complications. You just have to take like a no knowledge approach and use a really weak statistical test, like a rank test that makes no assumptions about the underlying distribution of the data. And that is a problem that you kind of need to know is a problem before you can do it right, and otherwise you end up with nonsense for your statistical test. If you're comparing treated versus controls, it's simple. You would think it would be just a simple test. You can use a t-test, no problem. You can't. And I'm going to make the assumption that metabolic markers are very similar and that every single one is a special snowflake on its own. And you cannot make assumptions like that. So if you want to build a good statistical test, you also have to build a separate one for a big metabolite. Something like that.
Yes. A lot of fun and powerful data set is how you should rephrase that. But actually, that's a good point. So, Paola, you are involved in drug discovery, right? You are identifying targets, you're moving drugs through the pipeline. You have access, either internally or through partners, to do these omic technologies. How do you evaluate them? Like, is it true on your end that you think genomics is just table stakes, and then you have to like make more justification for proteomics and metabolomics? Like, how do you evaluate them?
Yeah, I think we use the different data sets for different reasons. For example, going back to what Andrew was saying, I think where maybe a genomic signature might be very, very useful is when at the beginning of the drug discovery process, we prioritize which targets are genetically associated with diseases. So that could be where genomics is very powerful. Then I would say once you start to develop the drugs, we have to keep in mind the drugs really need an enzyme, really need a protein. So there is only so much that you can learn from a transcriptomics or a genomic signature. And I have experienced over the years that I've been working on proteomics a true change into the mindset of scientists with the advent really of these new modalities, the protein degradation modality, because then people really started to look at the protein degradation. So you cannot use transcriptomics, you really need to measure protein expression. And that has been, in my experience, a changer situation. Even I was within the AstraZeneca organization at that point, how now proteomics started to really expand. Now many companies actually have proteomics departments, and together with chemical biology, which is part of my background too. So these different steps of information come with different knowledge that then you apply at different stages of the project.
Yeah, I think that's great. So you said something again when we were speaking before that you think there's like a sweet spot where these different omics have value. Yeah. And like, so I've always been like, start from the beginning and integrate omics. It sounds like you probably disagree that sometimes it's actually, you gotta move things forward a little bit more. Is that right?
So maybe I'll speak more closely to my personal experience with proteomics, because I've been thinking a lot about when it's too early to work to characterize a molecule with proteomics and when it's too late. So for me, the sweet spot has been between lead identification of a new molecule to candidate formation, because that is the sweet spot where you need to understand the mechanism of your drug, you need to understand the selectivity, you need to define the liabilities before you declare a clinical candidate. Because once you have a clinical candidate, you need to go into, your IND filing. You want to go the package where you are very convinced about the mechanism of your molecule. You don't want to discover things later on. And so, but also you don't want to generate this knowledge too early because the chemical pharmacophore will change. So how that is the sweet spot. And then regarding the integration, maybe early on, even as a maybe lead to the campaign, understanding transcriptomics, I would say, together with genomics, even to compounds to intervene in biology, not so much to characterize your drug.
So, Eleanor, I don't know if you can feel about that or what you're seeing from some of your clients. Are you seeing maybe the genomics and transcriptomics as earlier projects and then proteomics more?
I would say so. I mean, it depends on the modality that they're going after, but yeah, one of the things people use transcriptomics for most is mechanism of action studies and that kind of off-target effect assessment. Really, really common to do that. The other thing that they're using, single-cell RNA-seq technologies are not really useful for figuring out if your drug is actually affecting the cells you care about, which is really important, and if they're messing on other cells. So yeah, and then proteomics, people get into proteomics when they're impacting things like splicing a lot too, and that can happen really early on. But I would think most of the time that happens later.
Not that or you or even maybe Andrew coming from the other side. So like are you ever seeing genomic customers or clients that are more established, right? That come after I don't know, the target's already figured out or stuff like that, or what do you guys think?
Yeah, a lot of part of the CP tag initiative of the NCI is that they initially targeted samples that have a very strong genetic profile that's been characterized. Once they know what the genomic profile was, then they did proteomic analytics on it, and then they went for deep proteomics research. So having genomics as a base is fundamental to do good proteomics, and then correlating the data to transcriptomics to have a protein genomic.
It makes sense to me. What the one of the fundamental differences though between genomic-based studies and what's come to us is, the size of say whole genome data is just so massive, and if you don't have some sort of idea of what you're looking for, it's basically useless because for the clinical applications of whole genome sequencing, right? I mean, that's why it's almost impossible to get diagnostic clinical whole genome validated through, approval because you need to have 20 PhDs in all these functional areas to understand what you're analyzing. And this is a different space than what we're talking about, but it just goes to show how much data there is and how to handle it. So I would agree with you that most customers and most core groups that want this type of analysis already have a backbone. They know what they want, that they want all the data just in case. Yeah, that's what we're hearing, rather than, we just want these 20 targets or 50 targets. If the order of magnitude between the cost for 25 targets and whole genome sequencing is coming closer together now. So why not just do a whole genome, and you get everything. So
Thanks for your buck as well.
absolutely.
So that brings up an interesting topic, that with these omics technologies, now we are able to move away from hypothesis-driven science and just measure it. And do we find something? And I'm curious for the entire panelists, like, what do you guys think? Is that good? Is that bad? What pros or cons can come with it?
I think that's a terrible idea to start with.
Let us know why.
I think science in general and experiments in general should be hypothesis-driven. There is absolutely no point in generating oodles of data, gigabytes of data. If you can't do meaningful statistics on it and draw biological inferences. What's happening a lot, especially with this is happening initially in genomics and now to a certain proteomics. A lot of the scientists coming out are technical experts. They know how to manage the instrument, service the instrument really well, and understand their little field really well. But to get an understanding of the overall biological picture, experiments are not always hypothesis. And that becomes a problem because you're generating a lot of data, and if the data is not driving a hypothesis, it's just meaningless. And one of the problems with proteomics is it's not as mature as genomics, is that it takes time to generate enough data to do meaningful statistical analysis. It becomes a problem that you cannot draw biologically important inferences. For example, if you have a protein which is upregulated or downregulated, if you're looking at only 10 samples, you don't know that that protein that you see is a differential expression. Is it because of batch effects? Is the system wasn't working well? Or is it biologically relevant? So the goal of proteomics, and that's what Bruker is pushing to be high throughput, just as genomics is, so that we can do population level proteomics like the genomics is doing.
I may disagree with you slightly, a slight nuance, but I want to hear from everybody else.
Yeah. So I just want to make a couple of points. So one is about these hypothesis driven. I just want to clarify that in my opinion, all the proteomics output that we generate, they are also hypotheses that then we will need to go in the lab and validate. These are all biological hypotheses that we generate. But what for me is very, very important as we need to do these large proteomics studies, for example, is to have in the room, as you were saying, understand the question that you are trying to answer. Design the experiment, the key biologists that understand what are we trying to get out of the experiment. We need to have the bioinformatician, the statistician in the room, because otherwise they will give you half time. Go and analyze.
Why didn't you do one more replica?
Yeah. Perhaps you spend 3 months designing an experiment, doing an experiment, and then they cannot analyze it, or they refuse to analyze it. And so that's very, very important to have a team. I mean, proteomics, when people think about building a proteomics team in a company, you just don't need a machine and a technical expert that knows how to run the experiment. You need a network of expertise and people that talk the same language to get the output. Because otherwise, you just generate a bunch of data that are meaningless. And then people get afraid, oh, I don't want to even generate a phosphoproteomics data set because I don't know what I'm going to do with that. So we have to be careful.
I just want to reiterate, call your bioinformatician. You're waiting on the phone. We really love designing experiments. It's one of the best things that you can do. It's really worth doing. And I agree, it's better to have a hypothesis when you go into designing an experiment, except in very special cases, because it forces you to actually think about the biology you're trying to study and design something that will actually work. Because people really do just throw things against the wall. It's like, oh, it's the genomics. It's going to give us the answers to everything, and it will not unless you ask the question properly. The exception that I want to bring up is there are some people who are designing big, cohesive, rigorous, systematically collected data sets for particular given like exercise that is kind of hypothesis free, but kind of not, where they're like, this is our therapeutic approach, this is what we're looking for. Those things can be gorgeous and are totally worth doing in a way. That's like real project.
I do have one question then as well, in terms of sort of thinking about it. I mean, the difference between the reductive, heavily reductive science, and one question, one lab experiment, one outcome, when people are trying to connect, many years ago, trying to figure out literal pathways, versus what we have now, which is a different paradigm. We have magnitudes of data, and I guess the question is whether or not then it's, thinking about machine learning, AI, unsupervised learning of tons of data sets where you don't necessarily need a biological relationship between these different data sets, and it's not necessarily required to find relationships that traditional statistics cannot find, and are not really meant to be, to be used for. So I'm just wondering what, maybe for you, what the usefulness of that is. Designing machine learning algorithms and then just kind of letting it go and seeing, unsupervised what it's finding and whether or not if it's finding something, is that useful, or is it just, of course it's going to find that,
There's a bunch of questions in there.
Yeah.
So you can absolutely go find a public data set like PCGA and then go do some data mining in it and find things. Probably a bunch of other people have found it. Not new stuff there. But we do data mining on existing data sets, public data sets, publications, things like that, and we find useful bits of information for our clients from that. Almost none of them are big enough for an AI approach. And an AI approach is as useful as the quality of the data that you put into it. I would argue PCGA is actually not good enough for most deep learning projects. But you can do some traditional machine learning. That's how I describe things that most people just call statistics. So linear models and things. Deep learning is really designed for something that is rigorously cold-fed. It works really well on rigorously cold-fed data. Image data works really well. This kind of data that I describe people creating as the focus for a startup. Their job is they're going to generate millions of single cell profiling profiles from perfectly curated set of cells run in perfect conditions in one lab, in one location, with one set of tests. And they're going to use that as a basis for their machine learning. They're going to have a team build that understands the biology of it. That can succeed. But cobbling together multiple data sets from many places and throwing it into a generative learning environment, I don't think it's going to be real successful. I think people that they should try it. And you can usually tell really early on if you're going to get something out of a data set. If a traditional machine learning method, on a set of 50 samples doesn't have anything, then deep learning for. Did I answer the question?
Yeah. Certainly also as well, when you're talking about like supervised learning, for example, for classifications, you have to have the most important thing is good outcomes data or whatever you're classifying. But I was just thinking in terms of the word omics to me not necessarily, but in my mind it just means a lot. I think of a lot of data, and I think of ways to usefully using ingenuity as well, take that data and make something of it, something that we may have not had the ability to do before we had some of these newer techniques. Statistically.
I think the reason I was going to slightly disagree with you, I agree with all the points you need, like good quality data, and I think the link to biology is an important one for me. But when I think of some of these big initiatives like the All of Us project or these big biobanking efforts, I think it's under the assumption that we don't know what to measure yet. That I truly believe there are biological markers that explain why somebody gets Parkinson's and somebody doesn't, and somebody responds to a therapy and does not. I don't think we know what all those markers are yet. And so I think setting up initiatives that let us in the right statistical fashion and with confidence find them instead of just like hodgepodging, together, a bunch of things, I think is really, really important. We're about 35 minutes into the conversation. I want to pause here. I have a million more questions that I could ask, but I would love to see if the audience, yes?
Yeah, so I'd like to follow up on what you just said.
Sure.
So before I put DNA into animals and I could follow the DNA, and that's nice because you start with what you know. Now I'm thinking about a disease. And it's a disease that babies get. Some of them get it, some of them don't. There's no rhyme or reason, all right? So now, which of your omics am I going to use to be able to identify the babies that are going to get it? Right? So, I'm kind of omicked out here because I can't, the idea that you could look for the differences in RNA, maybe there's a lot you could do the comparison. Yeah. Or you could look at the different proteins that are made. And I want to look at baby poop, and I want to be able to identify which babies are going to come down with the disease. So that's a challenge.
Yeah, I don't know that we know the right omics. I'll just say, inborn errors of metabolism was one of the great showcases for different omics, right? That if you have a mutation in this particular gene, you cannot make this enzyme, which thus you cannot make this metabolite. But
These are premature babies, and it's not, there apparently is no genetic marker to it.
Well, I will say too, though, over the last 5 to 10 years, with the advances in whole genome sequencing and the amount of the actual genome that we are really getting, because when they say whole genome 10 years ago, it's not the same as it is now.
Right.
In terms of the actual breadth of what you're getting, things like large structural variants, microsatellites, expansion repeats that are hidden. There's an entire field now literally dedicated to building the best reference genome.
Right.
Literally, the NIST people, Peter Cruz, Justin, so these guys are literally creating unbelievable new iterations of it, and so there's definitely some advances in these sort of hidden.
Speaking as a DNA person, that's
from the DNA perspective. That is purely, but that's a foundation.
Right.
That's where you start.
Sure.
Clinical whole genome or clinical whole exome is where you would start at Mass General with babies that failure to thrive, and they need a turnaround time in 2 days, 2-3 days, so that they can immediately start treatment or to try and figure out what the best course of action. That's like in many cases, like the first stage game.
Thank you.
I think your question actually calls up the need for really need to take a biological approach rather than a data first approach for something like this, because I want to ask you 1,000 questions about these babies. Like, is there a geographical connection? What does the group look like? Is it different? Right? And you can't just start with omics for a question like that. And if you did, I would say go from cheapest to most expensive and just do the genomes and then do the transcriptomes. And, if you're fishing, but like you won't get there just going technology.
So the question is whether the transcriptome would give you information, the same information as what the proteomics would get. Because you're going to make the assumption it's made in the RNA, and you'll be able to look at the differences in the RNA as more.
I think the scientific trend or acceptance is that there is about a 70% correlation with what mRNA is studying compared to what proteomics is studying. But there will be cases where there is no correlation between the two. So this is where the need for perhaps a multi-omics approach comes. And the other complication that I would like to add here is depending on the input material that you use, the different lines of the different tissues, the same omics will give you a profile that is different. So I think we should maybe shy away from thinking I'll do a multi-omics approach that's my answer, because it's rather, there are so many nuances that we need to take into consideration. And it's sort of mechanism of action is not an easy thing for identifying the change that is correlated with things. The omics is a tool that will help us to get there, but in a very systematic approach, an experimental approach.
Yeah. Any other questions? Ernie Schaefer, have you ever not asked a question?
My comment is to you, I mean, in clinical medicine there, we have the expression, there are many beautiful hypotheses destroyed by ugly facts, number one. And number two, there are many major discoveries that have been made by chance. Just one thing. Yes.
All right. Oh, so yes, please.
When you're setting up all of these totally complex, you're collecting a lot of data to feed the workflows and to theoretically train and retrain the workflows and models that you're building. How important is it to have a brand unifying metadata organization process in the beginning versus just saying, I don't need that, I'm not going to set up these multi-omic atlases. I'm going to take this data set and you know, this specific information, and I'm going to focus on the data associated with that particular aspect of anything that I'm working on. And I'm going to make sure that that's rigorously structured, and then I'm going to run it through my mind. Because to me, it seems like there are two different schools of thought. One is you got to organize the whole omic data around the world and make sure that everything is universally organized, versus there's too much stuff, just focus on what you're going to do, make sure that's clean, get it into the workflow, and analyze it.
So literally my next question was like about like getting to basics of how would you actually like set these up, right? If you're doing like a single omic study and a multi-omic study. So I think it sort of touches on that. And I have ideas, but maybe Eleanor, going to you as well, like in the hot seat here.
I mean, I was just going to say it depends, because obviously there's lots of different ways you can approach that. It depends on what you're doing next, right? If you're people like molecule and you don't have any money, obviously you're not going to invest in infrastructure. You're just going to do it. If you're a big company or you want to be a big company and you're planning ahead, you might invest in a real infrastructure that will support all the data types you expect. But I think you have to be careful that you never know what data types you're going to expect. So sometimes it's really hard to plan that far ahead, and you want to be really flexible. And sometimes the simple system is the best one to start with.
Yeah, I completely agree. I mean, so what you said also brought to mind another area. So there's like what individual users or companies do, right? How do we set up our metabolomic study? And we're setting up a collaboration with genomics, right? So how should we integrate our data? Should we run them in parallel as like separate studies, or do we integrate first and then do statistics? Because that will change the dimensionality of your data and thus change a lot of sort of different things. I personally don't know that there's a right answer. I think, yeah. But what I will say is some of the reasons that genomics, I think, has leapfrogged forward over the other omics technologies is because of some of the standardizations that exist. Like, you can't publish a genomic paper unless you deposit the data and you describe how you did it. Metabolomics data, and like Amy and Leo, look at you, I feel like it's a mess in like the public domain. You know, like our investors used to ask us, like, why can't you just go mine, you know, publicly available data to find a metabolite biomarker? And it's impossible because either results aren't reported, how they collected it isn't reported, or it's just not clear. So I do think there is benefits in setting sort of best practices and standardization policies. And in the metabolomics field, like we're going through that kind of like reckoning now where leaders like here are trying to sort of put those together. I don't know if you see the same thing.
Yeah, that is what we have a consortium right now called and Proteomics MQA Proteomics.
Call it MQAC. What does it actually stand for?
So that is what we're trying to do, is to establish the foundation of what is the best practice. At least when you report data, what's the basic thing you should have. With mass spec, which I think we have a couple reference published already. But with NMR, it's really tough. It's really tough. So we're working on that.
I have a question regarding the standardization is one thing, but what are the main areas you see under Omics will get you in clinical diagnostics as a from genomics to metabolomics to proteomics, like looking at special omics, but also in a multi-omics approach? What are the main areas you see?
You're a clinical diagnostic, yeah. Okay. There's a lot from regulatory to reimbursement to actually just like investors funding those types of companies, right? So diagnostic companies typically, you can speak more to this, you know, don't get reimbursed at the same level that therapeutics companies sort of do. So you spend all this time, you do this super powered multi-omic study, curated right, multiple centers, everything's collected beautifully. And at best, you might be able to charge a couple $1,000s for that product, right? And so, but you would have had to spend millions to sort of get there. So it's a huge barrier to just even create the incentive to collect that data, to sort of do it, to uncover sort of the biomarker. And then, the regulatory proof that is needed as well, you need to show analytical and clinical validation for each one of those omics that you're touching. And again, Ernie and Paola here who run clinical diagnostics labs, you need to, for every instrument, you would have to show that it works and run all these validations. Do you want to jump in?
No, I agree with you.
Yeah, it just creates a huge burden. Now, I think that's where we're headed. But I think we're going to need some changing in both regulatory and reimbursement frameworks to even make that a possibility.
In terms of the clinical diagnostics space of all these genomics, talking most established in terms of the regulatory framework around them, around what is required for like analytical validation, performance of your test, and then extending that to the clinical side of things. Just for example, in standard genotyping, if you want to bring out a new test, some new snippets associated with X, Y, or Z, you have to both establish the analytical validation and the performance, which is easy now because it's been around for a long time. You know exactly what you got to do, how many samples you have to run, what you have to validate, what the calculations are, how many days you got to run. So it's straightforward. Then, however, for the clinical aspect of it and what your interpretation of the actions that are recommended, that either has to come from a study that you run or from robust literature. And you have to compile all that, write it up. The caveat is that in the current regulatory space, and this is come up in the Ballot Act, but in the current regulatory space, CMS is the oversight. For us, it's a CLIA CAP, College of American Pathology Lab. You establish all of that, you write the validation, then you can start running tests. There's a little bit that goes more into it than that, but anything that is correlated or has the 3 letters of FDA tagged to it is a completely different story. And the last caveat would be New York State. New York State in the clinical space is exceptionally difficult because in order to test any, in clinical laboratory for diagnostic use there, you have to have the test approved, especially in the genetic space. So it's just sort of that's you.
Yeah, I'm going to raise my hand. I'm Pat Nolan, president of Clinical Enterprise. I've been in clinical diagnostics for over 30 years. The reimbursement challenges are a real constraint to diagnostic discovery because you can spend 5 years developing something and you get a CPT code and they value the CPT code X. That's only as good until they say it's not good. And so not only are you doing all this work, maybe a couple of like TrueGraph, TBI, for example. They could wake up in 2 years and CMS lowers the reimbursement. All the private payers tied to Medicare reimbursement, so you can never count on your revenue. And so it's really a big constraint in clinical diagnostics, which is it's hard to make investments when you don't understand what your runway is going to be at a certain reimbursement level, and it's a real problem.
I think the science is not the problem. I actually think it's a business framework that needs to be solved. In the last minute, and then we'll leave some time for networking. I have 2 questions for you, panelists. Andrew, we'll start with you and go down the row. What do you think is holding back omics technology? And what do you think omics could solve in the future?
Well, from my perspective, what's holding back omics technology overall is the expertise coming from your perspective of the infrastructure and engineering and foundation to set it up. There's a big barrier, I think, learning curve barrier for traditional scientists or even just straight data scientists to integrate those two. So infrastructure is a ton of money. Someone asked a question about that. And, is it cloud storage? Are you on-site storage? Is it hybrid storage? These are all things you also are thinking about, like, not just the science. Number one, that's number one. And number two,
But what do you think you could solve?
A lot.
Okay.
I think just in the genomic space, as a small example, it has expanded our understanding. And what I can tell you is that genomics and what we thought was sort of table stakes is not actually. There's a lot more going on, and so I think if anything, it can light new fires under ones that are burning out. We thought were burning out.
From a proteomics perspective, I think it's, as somebody pointed out over here, mass specs are still unfortunately very expensive. You need a lot of specialized talent to do these experiments, and because there are so many flavors of it, comparing data from one lab with one set of mass specs, one brand of mass specs, comparing it to another with another set of mass specs, another brand is very, very tricky. The cost of doing experiments is high, so we can't do large sample cohorts at a reasonable cost. Throughput is relatively low compared to genomics. So what mass spectrometry-based proteomics needs to focus on is lowering the cost, lowering the difficulty of use, and increasing throughput. I see that we're going to get there in the next 5 years, and when we do, we will be at the same state, not technologically and data-wise, with genomics. Then we can really drive the proteogenomics space and answer the questions that remain unanswered by genomics.
And any prediction what proteomics will happen?
I do not get any predictions.
Okay.
Right.
Well said. Eleanor, again.
I actually love the idea that proteomics might be a similar cost to genomics. That would really change the landscape. I interviewed for a job once at the Broad, actually, and I interviewed like 15 people on the same day. Wild, and one of them asked me, what would you change about, the biology technical landscape? And I said, well, I would really love proteomics to actually be as easy as transcriptomics. That would be awesome. He was like, wow, I'm a protein biochemist, so that was a really good answer. But I still think it's true, because what are we looking for in the RNA except for some kind of signal about what's happening in the protein level, right? So that would be amazing. The thing that I'm excited about personally is actually spatial transcriptomics, where you can look at tissue samples and see the transcriptome right there in the tissue. That's going to be a game changer, I think, for especially discovery biology and some diagnostics.
Do you think like any area that Omics is going to like help solve a testing problem or all of them? Great answer. Great.
I think predictive cancer research is going to work. And that's where the spatial biology comes in, like taking tissue section and finding out what certain proteins are upregulated and downregulated, and in the large scheme of things, how the interplay is going to be critical in understanding how tumor regeneration.
Yeah, I started with the positive. Yeah, what are we going to use these omics for? I think it's to develop better drugs like precision medicine, so that when we understand the disease, we can really treat the right drug, that we start with the right enzyme and the right population. And I think the reason why we're not there yet is because I don't think we have the tools set up because of the cost, because of the throughput, and I would say also because the data integration is in a challenge. Because sometimes we try to integrate transcriptomics and the proteomics by looking at the same time point. We don't yet understand what is the best way to integrate multiple data sets. So that's what
No, I think that's great. I agree. I think Omics is the way that precision medicine becomes a reality. I also think it helps open, like you said, new areas of understanding and like parts of biology that we thought we might have figured out, but maybe there's like new lessons to sort of learn there. So I'm just going to end next slide with a couple of thank yous. First and foremost, thank you to this amazing panel here. I sent this cold email to everybody and said, hey, we're trying to do this thing. Would you guys be interested in being a speaker? And thank you guys all for participating. This has been really wonderful. Thank you all for coming to our first event. We are going to be hosting several of these throughout the year. And we have support from the landlords here, Outshine. So Outshine helps provide this lovely spread over here, which when we break, I hope that everyone gets to partake in. And we also have support from the city of Framingham. The mayor's office is here. So we're really trying to do our job or what we can to help build a great community, to bring sort of people together and for those aha moments to happen. So I will end with asking the Olaris team to stand up just very, very quickly, because we are a very small team that is doing a lot of different things. And when I asked everyone to take pauses in their data science and their machine learning and in their mass spec and in their NMR to put together a seminar, I feel like people could have told me to buzz off, and instead we learned how to kind of set up mics and put all of this together. So thank you guys all, and just thank you so much.
The parking down here is great too.